Welcome to Let's Talk, straight talk, hard topics, smart people. I'm Stephen Graves, and I'm here at the SNMMI conference here in Toronto. I'm talking with Dr. Robert Hobbs. Welcome, Robert.
Thank you.
So you have a background in nuclear medicine and now a practicing radiation therapy physicist. I have a background in radiation therapy. I'm now a practicing nuclear medicine physicist. So we kind of switched in our roles in some way. So the question is about what can we learn from radiation therapy in terms of normal tissue dose-effect relationships and tumor dose-effect relationships. We know that the regulators have sometimes enforced artificial limits on normal organ doses for safety reasons and trials. Is that appropriate, or how do we extrapolate, and to what extent should we extrapolate?
Wow, that's a very loaded question.
And as you know, we discussed this recently at a workshop on the East Coast with FDA and a number of other important stakeholders.
So I think we need to be careful. I mean, I think it's always good information. I think what we can certainly learn from an external beam is that we need data in order to really establish, normal organ tolerance thresholds, which we don't have, or we have very little of in radiopharmaceutical therapy. So that's what external beam did. They had a whole bunch of data, and then they can, compile it and understand. Whether we can take the values directly from external beam and translate to radiopharmaceutical therapy, I think depends a lot on the radiopharmaceutical. And we need to stop thinking about radiopharmaceutical therapy as a single entity and saying, we're taking this value and we're translating to radiopharmaceutical therapy, because there are multiple radiopharmaceutical therapies and the thresholds could be very different. Now, in theory, we like to believe that radiation is radiation, and at some level we should be able to make the equivalence. The problem is, as you know, that the method of delivery is different. In external beam, it's driven by geometry and anatomy, because we direct the beams, and of course the toxicity is proximal, and the dose distribution in normal organs is not uniform at all, right? Whereas in radiopharmaceutical therapy, to first order, it looks uniform because it's driven by physiology and uptake,
And it might be with a high energy beta.
and it might be, well, the uptake again,
Depends.
you're going between activity and absorbed dose, right? Because even if the activity isn't entirely uniform, and it might be uniform, even if the activity isn't entirely uniform, again, as you say, if it's a high energy beta and the range of emission is long enough, then the absorbed dose ends up being relatively uniform. But that doesn't seem to be the case very much with the kidneys in particular, which are often the normal organ at risk in radiopharmaceutical therapy. I think we have better luck actually with the bone marrow, even though it's a more complicated normal organ because it's systemic and harder to measure. But there aren't that many. If you have a drug that goes directly and affects the bone marrow directly, then it's never going to work, because the threshold for tolerance of bone marrow is so low. And so the dose from something that is in the whole body is probably is only coming from cross dose, and probably not much from self dose. And so that translates reasonably well. But what uptake in the kidneys is generally localized. And we talked about this. The issue is how good an approximation is that? And I think and also in the RPT Tech group, which is a committee designed to study normal organ tissue tolerance in an analogous way to the high tech and the quantech. The idea is, let's just use whole organ dose thresholds established by the individual radiopharmaceuticals. And we think that, especially the ones with the long-range betas, they should probably be somewhere near the values of the external beam.
When accounting for dose rate effects.
When accounting for dose rate effects, yes, exactly. Excellent point, because that is the one thing that we can agree on, that we do need to account for dose rate effects. That will narrow the distribution, whether it's at the small scale or at the whole organ level, for sure. And then the ones that are much more difficult, like the alphas, because not only do they have the short range, and so the localized uptake of activity is going to be resulting in a localized absorbed dose, but then also you have RBE factors, which are unknown and may depend on the different source and target compartments within
the subkidney localization mechanism, whether it's bound to a PSMA receptor or whether it's in the proximal tubules or radiating the glomerulus. The microdistribution matters quite a lot.
Right. So those are much more complicated, and we really don't have much of an alternative than to determine what the normal organ tolerance is for each radiopharmaceutical therapy independently, just looking at the whole organ, because the whole complexity of how to translate down to small organs is a project that we are working on, and we hope to, bring to the table at some point. But and so then that would be nice is then a dose will really be a dose will be able to equate, and so that when you combine between an alpha particle RPT and external beam, you will be able to relate the values that you have as significant to potential toxicity in external beam as well. But for now, we're not there. And so the normal organ values that we're going to use on an organ level in radiopharmaceutical therapy should be established independently. And the physicians made an excellent point on this as well. They said, we don't want to use external beam values because they're not relative to our patient population. Even if they were the same absorbed dose values, they don't represent the lifespan.
That has everything to do with the toxicity endpoint that the data describes, right? So, but I think one thing we can always agree on is that for a given organ, for a given radiopharmaceutical, for a given toxicity endpoint, there will always be a threshold below which effects are not really seen, and above which you have an increasing probability of seeing the effect and increasing severity thereafter. Well, we just simply don't know where that threshold is and what that response looks like in most cases for low energy betas, for alpha emitters. And so with this, RPT Tech initiative, I think it's a great start. But if you're trying to look at the Quantech as the parallel, I mean, they had 40 years of good dosimetry data to build on. Are we going to have to wait 40 years to get the right data for this? I sure hope not, because people acknowledge that this is a problem and are working towards it.
Yes. So kind of what we're doing is we're putting a point on it. We're saying this is what we would like to have happen. So we've written a white paper already. This is where we want to be. Now we're doing a literature search, and we're going to say, ideally, we wanted to go through the literature and establish a relationship.
Turns out it's a little thin.
Right, yeah, there's absolutely nothing there.
Yep.
In large part because we've been undertreating patients so much because of these artificial, external beam-derived values. And so we need more data. That's kind of the appeal we have. And so, but it's more than just an appeal. We are actively working with drug companies who are developing and who are the ones who are acquiring the data. And what we're trying to do is to gather all this data and archive it somewhere so that we can do the analysis and see what the values are.
It's one of those things that's a little tricky because every company in industry developing new radiopharmaceuticals would benefit from knowing what the dose-effect relationships look like in normal organs and tumors. And they each have a relatively small amount of data. But when pooled, you might be able to start addressing those questions. But there are real sensitivities to confidentiality in pre-approved products, right? So how do you kind of sandbox the respective data and still draw meaningful conclusions?
Right. Yeah, and then there's the other argument that I just said at the beginning that every radiopharmaceutical is different. So even if we're pooling the data, are we pooling data that...
So we really need to know what the suborgan distribution looks like, and it have to be very similar agents for it to be meaningful,
exactly.
Right?
Yes.
So I think that, we will learn more about dose-effect relationships over time. But how does that tie back to the uncertainty discussion that we had at the very beginning? So let's say you know generally that the normal tissue threshold for kidneys may be closer to 40 gray BED. What's a safe level to be treating at given our uncertainty? Should we sort of be in that low 30s range as a safety, or patients might benefit from that extra bit and you risk it a little bit?
I mean, I think 20% is for... I think our uncertainties on normal organs like kidneys or liver are definitely lower than on the tumor. I think we can think at about like 20%.
Yeah.
So, I mean, I guess you're right. I mean, 20% of 40 is 8. So should we be retreating to 32? I don't know. That seems a little low. Maybe the middle of the peak.
Yeah.
Like at 36.
Yeah, sure. And again, it depends on the risk benefit for the individual patient population. Some patients, unfortunately, may not live long enough to manifest those late effects. And so they would benefit from a more aggressive treatment up front. And how do you think about tumor dosimetry, meaning these are mostly metastatic patients, almost all metastatic patients today. When we talk about the threshold for having a likelihood of tumor response, which tumor? Meaning we have say a dozen tumors. Do you take the lowest? Do you take the one that is most likely to lead to health complications based on its anatomical location? What's your thinking on this? And I know that this is largely an unsolved question.
It is. I mean, that's why, one of the theories is you just treat to the maximum amount of tolerated by the normal tissues, right? Because it's just too complicated. I mean, it's fine if you have illegal metastases and you just have 2 or 3 and you say, okay, I can do, and again, they have to be large enough because we know that the uncertainty on the dosimetry increases exponentially as the size of the tumors get smaller. So if you have a few large oligometastases that you can think, okay, I can get maybe 25% uncertainty on, maybe you can include that into your dosimetry planning and say, well, if I get 120 gray on each of them, I'll be fine. But otherwise, I'm just a proponent of treating to maximum tolerated exposure.
Which I think is a common view among physicists. I mean, the concept of over-treating really only applies when you're curing patients. I mean, you can over-treat a thyroid case, right? Because you know that 150 millicuries will do, and it doesn't make sense to go to 400 for maximum tolerated activity in many cases. But when most of the time we're looking at life-lengthening treatments, we expect from a physics and radiation biology perspective, more dose is more cell killing, right?
The biggest bang the biggest bang you can give is the one that's going to benefit the patient the most. Is probably not an unreasonable assumption, I would think.
That's great. And on that note, I've been talking with Rob Hobbs, and this has been a great conversation. Thank you so much for your time. Let's talk again soon.